Good morning to all. Today I am going to discuss about citric acid cycle. It is otherwise known as GCA cycle or Krebs cycle. So in this part we are going to discuss about the introduction, the site of reaction, pathway, energetics, regulation and clinical importance of citric acid cycle. This is the overview of citric acid cycle where from glycolysis it is linked. Then there happens a Krebs cycle which can lead to an oxidative phosphorylation, and at last the ATPs are produced. So the introduction of citric acid cycle means Krebs cycle. It is otherwise known as Krebs cycle. It is discovered by Sir Hans Krebs. So here in 1937 this citric acid cycle was discovered and he got a Nobel Prize for this in 1953. It is also known as tricarboxylic acid cycle. Reduced coenzymes are produced and most important ATP producing pathway is the Krebs cycle. About 65 to 70% of ATP are synthesized. So the site of reaction. Here this is a structure of mitochondria in which the inner membrane, outer membrane, crystalline matrix are marked. Here the citric acid cycle takes place in the matrix of the mitochondria. It happens once for every pyruvate molecule in glycolysis, because in the second slide we have discussed that it is a linked reaction after the glycolysis. So the end product of glycolysis is pyruvate. From that pyruvate acetyl-CoA is formed, and from that molecule the Krebs cycle or the TCA cycle gets started. That's what here given takes place in the matrix of the mitochondria. It happens once for every pyruvate molecule in glycolysis. Purpose: conversion of acetyl-CoA to carbon dioxide. Generates reducing equivalents such as NADH, FADH and GTP to be oxidized in the respiratory chain to generate the ATP molecules. So here the overview of the Krebs cycle is given. The first starting material is acetyl-CoA, which condenses with citrate to form the continuation of the cycle, Krebs cycle. So here from where the acetyl-CoA is formed means it is from glucose, which converts into pyruvate and it gets converted to acetyl-CoA, and fatty acid gets converted to acetyl-CoA, ketogenic amino acid converts to acetyl-CoA, and after that acetyl-CoA, from that oxidation takes place, fatty acid formed, cholesterol formed, which is converted to steroid hormones, and the ketone bodies are formed. This is the overview of cycle where acetyl-CoA from where it is synthesized and what it gets converted to. So next slide, next portion diagram helps in this. Glycogen, which is converted to glucose, and that it is converted to pyruvate by the process of glycolysis, and a molecule of carbon dioxide is evolved and it forms a acetyl-CoA. Then again from fatty acids and amino acids acetyl-CoA is formed and it gets enters into the citric acid cycle where two carbon dioxide molecules are evolved. So steps involved in the TCA cycle. This is the first step. First step means it is a linking step from glycolysis to the citric acid cycle. So here the pyruvate, the end product of glycolysis, is converted to the acetyl-CoA with the help of an enzyme, pyruvate dehydrogenase. So this is the linking step of glycolysis and the citric acid cycle. So from that this acetyl-CoA enters inside the citric acid cycle and it forms the citrate. Acetyl-CoA condenses with citrate synthase to form the citrate. This is the first step, and citrate is converted to the cis-aconitate with the help of an enzyme aconitase, and cis-aconitase with the help of the same enzyme aconitase is converted to isocitrate. Isocitrate dehydrogenase converts isocitrate into oxalosuccinate. Oxalosuccinate gets converted to alpha ketoglutarate by the help of an enzyme dehydrogenase, and again alpha ketoglutarate is converted to succinyl-CoA with the help of an enzyme same dehydrogenase. Then the succinyl-CoA is converted to succinate with the help of an enzyme thiokinase, and succinate is converted to fumarate with the help of an enzyme dehydrogenase, and again fumarate is converted to malate, and malate is converted to oxaloacetate. So here the oxaloacetate is regenerated. That's why this is the circular diagram. So elaborately we can discuss here. So the first is step, which is a condensation reaction, that is acetyl CoA condenses with the citrate synthase to form citrate. Then the second cycle is dehydration, that is a removal of one molecule of water with the help of an aconitase that is converted to isocitrate, and isocitrate again it is converted to, in the next step it happens by hydration, addition of one molecule of water. Same aconitase is involved here to form isocitrate. Again oxidative decarboxylation takes place. There will be an evaluation of one carbon dioxide molecule, and again it forms alpha ketoglutarate. Alpha ketoglutarate, with the help of an oxidative decarboxylation, it forms succinyl CoA. Then again succinyl CoA is converted to succinate by the reaction named substrate level phosphorylation, and again dehydrogenation takes place to form the fumarate, and hydration takes place to form the malate, and again dehydrogenation takes place to form oxaloacetate. So this oxaloacetate is again regenerated, and two molecules of carbon dioxide is evolved in the cycle. So the starting step is the pyruvate molecule, which is a linking step where it is formed with the help of an enzyme. It gets converted to acetyl CoA. That acetyl CoA condenses with the citrate synthase to form the first step in the citric acid cycle, and later on, after all the reactions, it gets converted to the molecule oxaloacetate, which is regenerated, and two molecules of carbon dioxide are evolved in this reaction. So to prioritize, what is the purpose of a pathway? Here from this cycle we need to concentrate more in these steps. What is the purpose of a pathway? What are the starting and ending molecules? Where is this pathway? It means the site of the reaction pathway takes place. How does the pathway connect to other pathways? This is an intermediate pathway. It is connected to various pathways. See here, first we have discussed the pyruvate is formed from this fatty acid, amino acid, etc., and it gets converted to acetyl CoA. That's what here it means. How does this pathway connect to other pathways? Then what metabolic conditions turn the pathway on and off? What are the control points for regulating the pathway? That means control point means the reactants, products, and enzyme name of each regulatory step, additional regulatory molecules involved. Make sure you know every step that makes or uses ATP. Then what specific drugs or disease associated with this pathway? These are the things we need to concentrate more about the citric acid cycle. So next one, TCA cycle is an open cycle, operates only under aerobic conditions. So the previous section we have discussed about glycolysis, and there is two types of glycolysis in which two end products are formed. That is glycolysis under aerobic condition, it gets formed in pyruvate, and the end product in the anaerobic reaction is the lactate. So here there is no such type of reactions, it's only under aerobic condition. This is the final common pathway of oxidative metabolism. Two carbon dioxide molecules are released as a waste product of respiration. In this cycle I have first discussed that there will be a evolution of two carbon dioxide molecules, and the oxaloacetate is regenerated. Then here itself how much amount of pyruvate we are utilizing, that much times it will be repeating. So that cycle will be continued. Now for one pyruvate molecules, one time it will oxaloacetate will be regenerated, and again for the second molecule it will be again regenerated. So here acetyl CoA, oxaloacetate, FAD, NAD, GDP plus PI, it's all gives to 2 carbon dioxide, coenzyme A, oxaloacetate, FADH2, 3 NADH and GTP. Here energetics, 2 acetyl CoA from 2 pyruvates. So here 2 pyruvate molecules are generated from the glycolysis. Therefore from that 2 acetyl CoAs are formed. So acetyl CoA plus NAD plus FAD plus GDP plus inorganic phosphate plus water. 2 water molecules gives coenzyme A plus NADH plus FADH GTP plus 2 carbon dioxide. Here the value of the ATP produced, the number of ATP molecules generated are, for one NADH it means 3, and in newer terms it means 2.5 ATP molecules. For one FADH2, 2 or 1.5 molecules, 1 GTP, 1 ATP is formed. In generation of ATP in citric acid cycle, we can discuss number of ATP generated by oxidation is 3 NADH2. So here we have mentioned for 1 NADH, 3 ATPs are formed. So therefore, for 3 NADH2, NADH, 9 ATPs are formed. Number of ATP generated by oxidation of FADH2 are 2. Therefore, here 2, only 1 FADH2 is formed. Therefore, it is 2 molecules. Then number of ATP generated from GTP is 1. Therefore, it is 1. Total, it is 12. Then we have already said that in energetics, for 2 pyruvate molecules, 2 acetyl-CoA is formed. Therefore, it is the 12 molecules is for 1 acetyl-CoA enzyme. So 12 plus 12 or 12 into 12, it is 24. The total ATP molecules formed in the citric acid cycle is 24. ATP generation steps. Now we are going to discuss about from which steps the ATPs are formed. That is given in the old calculation and as per the new calculation accordingly in this tabular column. Here in step 3, where the isocitrate is converted to alpha ketoglutarate, coenzyme in that step is NADH2 and ATP is formed is 3. In newer calculation it is 2.5. And alpha, then next one, 4th step, alpha ketoglutarate, which is converted into succinyl CoA. The coenzyme involved in this NADH and the old ATP same as 3, and new it is 2.5. Then succinyl, then next the ATP generation step is 5th step, where the succinyl CoA is converted to succinate. Here the coenzyme involved is GTP and the ATPs formed is 1. Then the succinate to fumarate, the conversion of succinate to fumarate in the 6th step, it will produce as the ATP, and here FADH2 is the coenzyme, and therefore 1 molecule of FADH2 is equal to 2 molecules of ATP in older terms. In newer calculation it is 1.5. And in the 8th step, malate, which is converted into the oxaloacetate, that is the last regenerative step, where NADH is the coenzyme involved and their molecules formed is 3, which is equal to 2.5 in newer calculation. So as per this tabular column, the total number of ATP generation steps is 12 according to old calculation, and newer calculation it is calculated as 10. So these are the most important steps where the ATP is formed. ATP generation during oxidation of glucose. Net ATP production depends on shuttles used for the transfer of reducing equivalents from cytosol to mitochondria. Glycolysis, the number of ATPs formed are 8. Pyruvate dehydrogenase, it is 6, and citric acid cycle it is 24. So the total ATP generation is 38. Then significance of TCA cycle. Here we are going to say elaborately what are the significance of TCA cycle. There comes first the complete oxidation of acetyl-CoA. Here first we have discussed that is pyruvate is formed. From that pyruvate acetyl-CoA is formed. CoA enzyme is formed. So there will be a complete oxidation of acetyl-CoA in this citric acid cycle. That is the first significance of tricarboxylic acid cycle. Then second one, as provider of energy. This is the provider of energy, and we have already discussed that 65 to 75% of ATPs are generated by this cycle. And the third one is final common oxidative pathway. This is the final common oxidative pathway. And fourth one, integration of major metabolic pathways. That means nothing. It is an integration. That is different metabolic pathways comes together to form a product, same product. That is known as integration. For example, we have discussed there acetyl-CoA is formed from fatty acid or from amino acid or from pyruvate from the glycolysis cycle. So that all will be enters inside this cycle. So that is known as integration. Then fifth one, fat is burned on the wick of carbohydrates. Fat is converted, fatty acid is converted, and it is enters inside the citric acid cycle. That is known as fat is burned. Then sixth one, excess carbohydrates are converted to neutral fat. Seventh one, no net synthesis of carbohydrates from fat. Eighth one, amino acid enters tricyclic carboxylic acid cycle. Ninth one, amphibolic pathway, and 10th one is anaplerotic pathway. Now we are discussing about the significance of TCA cycle or Krebs cycle. So first comes the complete oxidation of acetyl-CoA. Here it is nothing but the complete conversion of acetyl-CoA. The steps which is included, that is acetyl-CoA condenses with the oxaloacetate to form citrate, and citrate is converted to isocitrate. Isocitrate which is converted to alpha ketoglutarate to succinyl-CoA. That is known as complete oxidation of acetyl-CoA. And the second one, ATP generation. Here 12 to 10 out of 10 ATPs are generated via substrate level phosphorylation and electron transport chain. And third one, final common oxidative pathway. Here it means from glucose in the process of glycolysis cycle, pyruvate is formed, and from tryptophan or which is converted to alanine and which is converted to pyruvate, pyruvate is converted to acetyl-CoA, and pyruvate is converted to oxaloacetate. From there the Krebs cycle starts and it is regenerated. So aspartate is also converted to oxaloacetate, and histidine, arginine, proline, etc. Which is converted to glutamate, and it forms alpha ketoglutarate. So this indicates it is a final common oxidative pathway. From various substrates, it is commonly formed inside this cycle. That's why it is known as final common oxidative pathway. And fourth one, integration of major metabolic pathways. Here from pyruvate, acetyl-CoA is formed, citrate is formed, oxaloacetate, aspartate is formed, and other amino acids, purines or pyrimidines are formed. From succinyl-CoA, porphyrins, heme, chlorophylls are formed. Alpha ketoglutarate, glutamate is formed, and other amino acids are formed such as purines, etc. And from citrate, it is converted to fatty acids and sterols. That's why it is known as integration of major metabolic pathways. This explains briefly about the integration of major metabolic pathways. And then comes fat is burned on the wick of the carbohydrate. For complete oxidation of acetyl-CoA, it needs the help of oxaloacetate. The major source of oxaloacetate is pyruvate, derived from the carbohydrates. So for the oxidation of acetyl-CoA, oxaloacetate helps, and the major source of oxaloacetate is the pyruvate, which is from the glycolysis. And excess carbohydrates are converted to neutral fat. Glucose is converted to pyruvate. Pyruvate is again converted to acetyl-CoA, and with the help of enzyme pyruvate dehydrogenase, and acetyl-CoA is converted to fatty acid. So excess of carbohydrates which is converted to the neutral fats. Then PDH is irreversible, that is pyruvate dehydrogenase is irreversible. Fat cannot be converted to glucose. This cycle cannot be reversed. That's why it is known as irreversible. And next one, participation of TCA cycle in fatty acid synthesis from glucose. Here starts from glucose. Glucose which is converted to pyruvate. It enters the mitochondrial matrix, and there happens the citric acid cycle where acetyl-CoA is converted to citrate. From the citrate, it is happens in the cytosol, and citrate with the help of an enzyme ATP citrate lyase, it is converted to acetyl-CoA, and it is converted to the fatty acid, where oxaloacetate is also evaluated. Oxaloacetate is relieved. So this is the overall participation of TCA cycle in the fatty acid synthesis from glucose. Then no net synthesis of carbohydrates from fat. Acetyl-CoA entering in the cycle is completely oxidized to carbon dioxide by the time the cycle reaches succinyl-CoA. So here first point itself we have said that completely this acetyl-CoA is oxidized. So when the step reaches the succinyl-CoA, it is completely, the acetyl-CoA is completely oxidized. So acetyl-CoA cannot be used for the synthesis of carbohydrates. Therefore, the acetyl-CoA is not used for the synthesis of carbohydrates. Eighth one, amino acid finally enters the tricarboxylic acid cycle. Here, first step and the introduction slides we have seen, it is an integration of many pathways. That's what here itself it is the meaning of that. And ninth one, amphibolic pathway. This is nothing but the cycle is amphibolic, means it is both catabolic and anabolic, where there will be a more substances which get break down, and there will be a joining of more substance or formation or synthesis of more substance. That's why it is known as amphibolic pathway. Then last one, anaplerotic role of TCA cycle. That means filling up reactions or influx reaction or replenishing reactions. Pyruvate which is formed into oxaloacetate, glutamate which is formed into alpha ketoglutarate, aspartate which is formed into oxaloacetate, pyruvate which is formed into malate. These are the examples for anaplerotic role. Regulation of TCA cycle. How the TCA cycle is regulated. Indicator molecules of higher energy state, that is ATP, NADH, citrate, acetyl-CoA, inhibit tricarboxylic acid cycle. So here ATP formed, NADH formed, acetyl-CoA formed, which will be inhibiting the citric acid cycle. And indicator molecules of low energy state, that is ADP, AMP, NAD, stimulate the citric acid cycle. Then inhibitors of citric acid cycle. Fluoroacetate, first one, it is an inhibitor of citric acid cycle. Condensation of fluoroacetyl-CoA with oxaloacetate which forms fluorocitrate, which inhibits aconitase enzyme, and accumulation of citrate will happen. Then fluoroacetate, which is a pesticide. Melanate, succinyl dehydrogenase enzyme. It is an inhibitor of succinate dehydrogenase enzyme. Arsenite, alpha ketoglutarate dehydrogenase enzyme, it will be inhibiting. And metabolic defects, extremely rare for this. Metabolic defect is rare for citric acid cycle. Defect in some of the defect may happen, means which is defect in PDH, that is pyruvate dehydrogenase. For examples of that are lactic acidosis, neurological disorders. Then second one, defect in pyruvate carboxylase, where the oxaloacetate levels are decreased, hyperammonemia, lactic acidosis, and hyperalinemia happens. Summary: pyruvate is converted to acetyl-CoA by the action of pyruvate dehydrogenase complex, a huge enzyme complex. Acetyl-CoA is converted to 2 carbon dioxide molecules via the 8-step citric acid cycle, generating 3 NADH, 1 FADH2 and 1 ATP by substrate level phosphorylation. Intermediates of citric acid cycle are also used as biosynthetic precursors for many other biomolecules including fatty acids, steroids, amino acids, heme, pyrimidines and glucose. The last one, oxaloacetate can get replenished from pyruvate via a carboxylation reaction catalyzed by the biotin containing pyruvate carboxylase. So here questions. TCA cycle is a final common metabolic pathway for the oxidation in foodstuffs. Justify. Conversion of pyruvate to acetyl-CoA. Energetics of citric acid cycle, Krebs cycle or TCA cycle. That's all for today's topic. We have discussed the introduction of citric acid cycle or Krebs cycle, and we have seen the site where it takes place, and we have discussed about the flow chart or diagram where the steps, which are the steps involved in this citric acid cycle and what are the significance, we have discussed elaborately. So that's all for this session.